Okay, well it's a privilege now to welcome back for the next talk Professor John Bryant, who's Emeritus Professor of Cell Biology from Exeter University. And John has not only done a lot of cell biology during his life, but has also done a lot in developing ethical and bioethical programs. In fact, I think started the first undergraduate bioethical course really in any British university, so it's been very much involved in the ethical discussions arising from biological discoveries over these past years. So we're particularly happy that he's come to speak to us now on these ethical challenge questions arising from modern biology. So thank you very much, John. Thank you.
Yeah, thank you, Dennis, for your kind introduction. And thank you, people, for coming back from a little break. Good to see you here. And this is your dinner's coming soon, folks. So well done on your stamina. Just as last night I suggested that it was some current topics in science and religion, and then Rodney covered some more topics, other topics in science and religion, and Dennis has covered more. You know, these are big, broad topics, and each of us has got our own particular interest, which we will develop in these talks. And so I can't claim that this talk is going to be an exhaustive review of the ethical issues in biomedical science. But we are talking here about a topic which is very much part of both science and religious and philosophy curricula. And when I go into schools to talk about these issues, sometimes I'm talking to A2 or AS geneticists, biologists, and sometimes I'm talking to GCSE or AS RE students. And so both subjects deal with this. And there's some quite nice linkages with what's gone before. You know, that Rodney has talked about the necessity for supernovae to form carbon. And then we heard Jennifer Wiseman talking yesterday about how humans have evolved and developed, and we have beings that are able, based on that formation of carbon, we've got beings that have evolved to be able to discuss these ethical issues. And Dennis gave us further insights into that a little while ago. And so I was reminded, before we start, of that song that was written for Crosby, Stills, Nash and Young for the Woodstock Festival, which goes, "We are stardust, we are carbon. We are stardust, we are golden, we are billion-year-old carbon." You know, she obviously knew, the author obviously knew a physicist. Actually, the last line in the chorus is even more interesting. It's, "We've got to get back into the garden." So there's a kind of combination there of old Earth and the Genesis story, all rolled into one. We are stardust, we are golden, we are billion-year-old carbon. And as I say, it's because we have evolved as societies with the ability to debate these things that these issues come up. I'm not going to cover very many of the current issues, and there are loads of them, just sort of reel them off. There's genetics, there's genetic selection, there's genetic modification, there's cloning, there's stem cells, there's nanotechnology, there's transhumanism, there's end-of-life issues. You know, all these have raised ethical problems for society at large, and we certainly haven't got time to look at them all today. So I'm going to be focusing on human embryos, genetic selection of embryos, saviour siblings, and we'll touch on embryonic stem cells and cloning. And I need to say that my own research expertise has been in the cell division activities of plant stem cells, so I do know quite a lot about how stem cells work, both in plants and mammals, but the major focus will be on these top two things, genetic selection and saviour siblings, which will become clear as we go on. But by way of introduction, I'd like to suggest that we've got kind of three threads that will run through the talk. And the first is a general one. It is this business of science and technology, and how should wider society make use of the findings of science, and what regulations are there on that? Who decides? Is that a general consensus thing, or is it centrally decided? And that linked with that is the fact that there are many things that science is enabling us to do, but the question is, should we do them? And you remember in Jurassic Park, both the book and in the film, the character played by Jeff Goldblum, one of my favourite actors, the characters he plays, and he actually asked the question, just because we can do something, does that mean we should do it in relation to the cloning of the dinosaurs? And it's a question that will come up time and time again as the biological power of modern science gets greater and greater. But thirdly, I think we need to challenge ourselves. If you have a religious faith, does it actually help you make decisions in this area? And I have debated with Christians who have a kind of gung-ho, knee-jerk reaction to these issues, believing in some way that the scriptures help them to decide. And my take on this is there is no direct guidance on any of these issues in the scriptures, and why should there be? They're not written in the 21st century. And therefore, if we believe our faith is helping us to make decisions and to arrive at conclusions, we need to be very honest in thinking about how that is. What is our faith helping us to do here? And so there are no easy answers. I love the Franciscan prayer that starts, "May the Lord bless you with unease at the easy answer." and that's what I feel about this, that people may have too easy answers. So I'm going to start with, if you like, a little mini history of human genetics. Human genetics has been really quite an active part of the whole field of genetic research since genes were first discovered or rediscovered back in 1900. And it had been realised, in fact, for generations that certain traits clearly ran in families. Some of those traits were physical. That there were certain people with red hair, for example, that trait would run in families. And it was also realised that some disease states ran in families without any understanding of why that should be at all, without the discovery of genes. And Professor Steve Jones, who writes extensively, an excellent populariser of genetics in the right sense of the word populariser, writes extensively about genetics and society, tells a story of a rabbi in AD 200, within the Christian era, who had circumcised a little boy on the 8th day, according to the normal Jewish rite, and the little boy bled to death. The bleeding didn't stop after the circumcision. And a little while later, the same family had another little boy, and he also bled to death. And the rabbi was wise and said, well, look, if you have any more little boys, we won't need to circumcise them to welcome them into the Jewish faith. We'll exonerate them from that rite. And said maybe we ought to include first cousins as well. And so he didn't know it, but he discovered excellent haemophilia, which we'll look at later. And as we've started to look at linkage and inheritance patterns, and then we've started to unravel the human genome building block by building block, we've got something like 4,500 single gene diseases. There may well be more. Some of them may be so rare that we haven't come across them yet. But approximately 4,500, where we can make a one-to-one linkage, and this is the key thing, a one-to-one linkage between a genetic change, a mutation of some sort, and they do vary, the sorts of mutation, and a disease state. And sometimes that is a 100% linkage. We call that 100% penetrance. If you've got that gene or that pair of genes, then 100% likely to have the state. In other cases, it's simply a high susceptibility, that although it's a one-to-one correspondence, it's not 100% penetrance. And so you have maybe an 80% chance in a lifetime of having the illness, such as, for example, breast cancer 1 mutation would be in that category, or 70% chance, the colon cancer mutation would be in that category. So, but they're nevertheless a one-to-one correspondence. But we've also got very much more complicated situations. As we've studied developmental biology, we understand that genes interact with each other, they interact with the environment, and they interact with each other in the environment. So we've got a number of different sorts of interaction. And it shouldn't surprise us then that our interaction with pathogenic organisms, viruses and bacteria, and with pathogenic factors, environmental carcinogens, is also subject to some genetic input. And so, we've all got colleagues, I'm sure, who always seem to get colds, man flu in commas or something like that. And they're not malingerers, they are genuinely less resistant to particular viruses. We know people, and I'm sure we all know them, who've smoked 60 a day since they were 15, and they're kind of 85 years old and still going strong. And yet we know of other people such as the trumpeter Roy Castle, who didn't smoke at all, but played in nightclubs where there's a lot of smoke, and got lung cancer from secondary smoking. And so there's a resistance then to environmental carcinogens. And Francis Collins, who we've heard quoted already, believes that there's some genetic component to all disease states, but it might be this rather complicated susceptibility thing. But nevertheless, what we need to know is that diseases were a major motivation for studies of human genetics right the way through from the start of the science. And I'll just give you some examples. We have the recessive illnesses, which I guess are the commoner illnesses. And here you need to have they're the commoner mutations, but they may not be the commoner illnesses, because you need 2 copies of the mutated, the changed gene, in order to have the actual disease. And so it means you need to inherit 1 copy from your mother and 1 copy from your father in order to be ill. And cystic fibrosis is a very good example for the Caucasian population of Britain, because in the Caucasian population of Britain, 1 in 25 of us carry 1 copy. And so if you work out the likelihood of having a child with another carrier, the odds may be a bit shorter than you'd like. And it is the commonest genetic state that's regarded as a disease in Britain, cystic fibrosis. And as we'll see later, the health authorities are trying to do something about that. It is based on a founder effect, that almost certainly it started amongst the Celts, and there hasn't yet been enough homogenization between the Celts and other Caucasians to make it an even spread. And amongst the Celts, and I come from a Celtic lineage, the carrier frequency is 1 in 22. So if you go to Cornwall, to Wales, Ireland, and so on, you have an even shorter odds of having a child with cystic fibrosis. But there's some very nice little examples of how genetics tells us a bit of history. In Iceland, the genome of everybody has been decoded. That's part of their health service. And they found the cystic fibrosis mutation is commoner than it ought to be in a Nordic race. And then you look at the history of Iceland, and they went down to Ireland to raid the slaves, particularly female slaves. And so that gene has gone into their population. And then another gene that's associated with a particular ethnicity, of course, is sickle cell anemia, where, again, both copies are needed for the full-blown disease. But having 1 copy of the gene does slightly damage your blood cells, but it also makes you resistant to malaria. So it doesn't surprise us then that the population, for example, of West Africa, and therefore of the United States of America, the black people in the United States, the African Americans, high proportion. So these will be recessive diseases, and the majority of genetic diseases are recessive. So you do need both copies, and sometimes the first thing that a couple knows that they're carrying the mutation is when a child is born with the illness. And that happened with some friends of ours around the time all the families around us were having our first children, and the first child of some close friends of ours had cystic fibrosis. And sadly, she died 2 years ago. And then you have the dominant, and here that you only need 1 copy of the mutated gene, and then you have the illness. And of course, provided there's only 1 parent carrying the gene, but each conception has still got a 50-50 chance of being unwell. And Huntington's is a very unpleasant neurological degeneration. I was in Cardiff before being in Exeter, and Cardiff is one of the centres for Huntington's research. It's a strange mutation. It's not a mutation, not a change in the order of the building blocks. It's an insertion into the gene of a piece of DNA we call repetitive DNA. And the repeat gets more frequent as you go down through the generations. And so let's say that somebody had it at 55. It's quite possible if they pass it on to a child that a son would have it in their 40s and so on. And so it works its way down. I met a Huntington's sufferer in Cardiff some years ago who was beginning to see the symptoms at 35, and his dad had shown them at 50. So this is a disease where the onset becomes earlier as you go through the generations. And then finally, the X-linked diseases. These are recessive. So they're recessive diseases, but they're located, the gene is located on the X chromosome. And I've already mentioned the classic one, haemophilia. This is Queen Victoria's gene. And so because women have got 2 X chromosomes, if you've only got one faulty copy, then you're absolutely fine. But men have got an X and a Y. And so if they've got a mutated haemophilia gene on their X, they've got no good copy to override it. So these are recessive illnesses. But the Y chromosome, have you ever seen them down the microscope? They're pathetic, aren't they? They do quite an important job, I would say. You know, they have the testis determining factor, and therefore they enable males to make testosterone and so on. But they have not got much else on them. Air guitar, I think, is one of the major genes. and football, perhaps, I don't know what else. Maybe an extra copy of the map reading gene, but they're becoming redundant and unexpressed as sat-nav takes over. So sat-nav may be depressing the map reading gene in both males and the extra copy, in etc. But anyway, the X chromosome is small. It's got very few genes on it, and certainly hasn't got this particular blood clotting factor gene. And so Queen Victoria, if you look at the male descendants of Queen Victoria, many of them died before they'd had children because they'd bled to death. And like the 2 little boys in the story I told you. Now, the Human Genome Project, its first work, as Dennis alluded to earlier, was to simply look at the order of the DNA building blocks in an average human being. And then it's expanded out to look at things like variation and so on. And variation between humans. And then as we've mapped, as we know where on the chromosomes many of these disease genes are, that's enabled us to start looking at the disease genes themselves and to unpick what has actually gone wrong. Was it a change in the order of the building blocks? Was it a deletion of a building block? Was it an addition of a building block of base? Was it an insertion of a piece of repetitive DNA? Was it the loss of the on-off switch, the promoter? And so there are many, many reasons why a gene may not work. And I'm calling all these mutations in a very loose way changes in the DNA. But the key thing is that we're getting a handle on many, many of these genetic illnesses based on our unraveling of the order of the bases and so on in the genome. And what that means is that we have very powerful diagnostic tools as we begin to add to our catalogue of diseases for which we know the genetic change. And the thing about a DNA test is you don't need the person in the room to do it. And so you don't need the symptoms. Now the symptoms, if you're already born and you're an adult, may well help. You know, so that, for example, if a new version of the Huntington's gene had occurred and somebody had begun to exhibit the symptoms in their 50s, the doctor would obviously look at the symptoms, but the clincher is actually the DNA sequence. And so we can go on and we can test children. Supposing you've got a little boy who's suddenly developed muscular weakness and he's no longer able to play football so well, he's getting tired. And the doctor suspects Duchenne muscular dystrophy. DNA tests will show whether that is true or not. Supposing you have a newborn baby and you want to check whether they've got phenylketonuria. Well, every single baby in Britain is tested for phenylketonuria. It used to be a biochemical test, testing for the ability to metabolize phenylalanine. Now it's a genetic test. Or supposing you want to see whether the baby's got cystic fibrosis, you can do that with a newborn. And of course in the film Gattaca, all the newborns were scanned. But also we can have babies in utero, fetuses in utero. You can get fetal cells from about 12 weeks. Unequivocally you can get them at 12. You can actually get them a bit earlier than that, but you're not quite sure that there's some maternal stuff in there as well. So you can test babies in the womb. But more to the point, you can test embryos pre-implantation. And this is the thing I want to talk about mainly. So we can move on to that in a minute. I'll just put out the code in DNA there over on the right, just to remind us. This is what we're looking for changes in the coding capacity, the coding ability of a specific piece of DNA. Sometimes the change is quite dramatic. You know, there's a disease called thalassemia where most of the gene is missing, and so you don't get any active product at all. So here we go. It's based on the remarkable developmental biology of mammals. These are And I have to acknowledge that this comes from a journal, The Biochemist, published in 1997, and they give me permission to use this in educational talks of this sort. So these are pictures of in vitro embryos. So in the top there you've got a newly fertilized egg, a single cell embryo, and although it may not be apparent to you, I can tell that the two sets of genetic material are still separate, and indeed they will remain separate. They behave as little independent packages of DNA for the first two cell divisions. So it's not until we've got 4 cells that the two sets merge to make one nucleus. So let's work out fertilization. That takes 10 hours before that's complete, and before the sperm is completely in there and unpacked and has left its genetic material in there. It takes 100,000 sperms to mount the cavalry charge to get one in. So it is just like in the Woody Allen film. unless you're performing it in vitro and you do ICSI and you inject one inside. 100,000. And as I say, the two sets of genetic material just behave separately, as if we call those two haploid nuclei. They've got one set of chromosomes each, and then they merge at about this point to form the diploid nucleus of the organism. And intriguingly, this is happening in nature in the fallopian tube. So you're not in the uterus yet. The egg is coming down, the sperm is coming up, and the twain do meet. And the fertilization occurs usually somewhere in the fallopian tube. And migration occurs down until this structure here finds itself in the uterus. That's called the blastocyst, and it'll become important to us later, about 7 days. But I want to pick up this point here. With the 8-cell stage, if you were a fruit fly, each of those 8 cells would have a definite function and a definite fate. And if you take one out, a bit of the fruit fly larva is missing. Whereas if you do that with a mammal, that doesn't happen at all, because the cells at this point have got no set fate, no set career at this point at all. And so you can take one out. Now, we need to remember that in Britain we've been handling human embryos in the clinic in these Petri dishes since 1978 or slightly before. I assume they had a couple of goes before Louise Brown was born. It staggers me that she's going to be 32 this year. She's had 2 of her own children, but they were born through normal intercourse. She didn't need to have IVF. So we've had a lot of experience. About 12,000 babies are born in Britain each year through IVF. And that's been a kind of running average for the last few years. So we've handled, the medical profession has handled a lot of embryos. And it was then wondered whether we might be able to use that knowledge and the knowledge of the developmental biology to take one of these cells out. And with the immense power that genetics has given us, and your biologists will have heard of PCR, a means of amplifying specific bits of DNA. One cell is enough, which is why rapists can be caught from one semen cell. One cell is enough with the power of genetics. And you can take a cell out of these embryos and test it for a particular genetic condition. And so there we are. This is a slightly old picture. Cell is being removed, and in this particular picture, which dates back to the 90s, if you like, the cell was removed by purely physical means with a micromanipulator and a tiny needle. But today, very accurate laser knife is used to cut the one cell out, and then it's sucked out gently. But the technique is the same. The picture is fuzzy. It's bound to be because the embryos became happy in a buffer solution, and we're looking down a bench microscope at it. You can see the picture, so that cell is going to be taken away. The particular bit of DNA we're going to be interested in is going to be amplified by a process called PCR, and we can look for a mutation. And so I'm going to tell you a few stories today. I don't know the real names in this family because they were never revealed. If you go to the HFEA, the Human Fertilization Embryology website, it lists all the diseases for which genetic tests are allowed, but except for about 4 of them. Some are so rare, they said, if we tell you that we're doing this, you'll recognise the families. The HFA protects the confidentiality of people who undergo this. So I don't know the real names because the family didn't go public. So let's call the little boy Matthew. But Helen and Peter knew, because of their family history, that they had cystic fibrosis in their families. And it turned out that they were indeed both carriers. Cystic fibrosis gene is an extraordinary gene. It's huge. It has many inserted sequences that aren't part of the original final code. very difficult gene to study. But nevertheless, by 1989, we had enough of a handle on it for this to happen. So Ellen did not want to have a child with cystic fibrosis. Remember, 1 in 4 chance for every conception. And so they underwent IVF, and the embryos were tested in exactly the way I've suggested. And embryos free of the mutation were then inserted into the womb, and baby Matthew was born free of the illness. That was 1989, so that was the first time ever that a human embryo had been genetically tested. And exactly the same year in the United States, the same test was developed for sickle cell anemia. So you have a common illness from two different groups of the population. And so from 1989 onwards, the story began to unfold, that more and more diseases came on stream. And I'm just going to tell you some stories about other illnesses to see how we react ethically. I hope you're thinking about whether you think that this is okay. You know, it was done out of, if you like, out of compassion for the couple, but some people feel uneasy about the idea that you test embryos. And if you feel uneasy about it, maybe you can pinpoint why. Maybe not. So here's Chloe. And Chloe is her real name because not only did the American family go public, but they had a film made of themselves, which is now available on the New York Times website. So here's Chloe. So her story is that in her dad's family, her dad is called Chad. These are their real names. And they're a Chicago family, wealthy Chicago family. In Chad's family, the colon cancer mutation is clearly present. And when you look at the pedigree, and that's when you map out the family genetic history, you can see succeeding generations, people have died of colon cancer. And the most recent person to have died of colon cancer was his uncle. And Chad has been tested for the mutation and knows he has it. It's dominant, so 50-50 chance of each child having colon cancer. Now, Colby doesn't have the mutation. Now, what about colon cancer? Well, I've said something about it already, but let's unpack it a bit more. This is a late onset condition. So Chad is in his mid-30s, but hasn't yet got it. And when you look at the age at which his relatives have got it, they haven't had the disease until they're in their 50s. So it's late onset. Whereas cystic fibrosis onsets from the moment you're born. This is late onset. So any child would have many years disease-free. And then I said the penetrance was 70%. Actually, in the States, the penetrance is 60%, and the difference is not really understood. It might be that the version of the gene we have over here is slightly different. But anyway, the penetrance is not 100%. So late onset, less than 100% penetrance. And then with modern methods of treatment, there's a 90% cure rate. So that's the etiology of the disease. That describes the disease that Chad and Colby wanted to avoid any child having. But they went through the whole stuff. They underwent IVF and embryo testing to avoid having a child with the colon cancer mutation. And Chloe was born, and in the video on the New York Times website, so if you go to newyorktimes.com and put in embryo screening video, provided it's up and running, sometimes it isn't, you can see the whole story in about 6 minutes. It's a very nice video. And Chloe is a little toddler there. She's obviously recently learned to walk. She looks like about 18 months old and is just beginning to verbalize. She's about 5 now. And the couple were deciding that when they had the second child, they were going to do the same thing. So there we go, a different situation. And again, I'm asking you to think about now, am I still okay with this? Am I still on board? Right, now here's the next story. This is the story of the 2 children. I don't know the parents' names, but I could find out because they also went public. So this is another American couple. And this is an American couple who lived in the state of Colorado, but actually went up to the north, to the state of Minnesota, where this particular treatment was available. It's Adam and Molly. Molly Nash. She was born with Fanconi anemia. Now that is a condition of the bone marrow, where progressively the bone marrow is less and less capable of making particular blood cells. And a child early on in life, like in the first couple of years, probably shows no symptoms at all, but the symptoms gradually get worse and worse and worse until the child usually dies. Usually between the ages of 11 and 14, it's that sort of condition, so young teens die of this. Now, because it's a bone marrow illness, you can imagine that you can do a bone marrow transplant, and that's absolutely correct. You could have a stem cell donation, a bone marrow stem cell donation, from a disease-free, compatible donor. And it's got to be those two things. It's no good transferring from a compatible donor who also has Fanconi. It's no good donating from a Fanconi-free donor who's not compatible. And you all know about graft rejection. And neither parent is going to be compatible because there's a 50-50, effectively a 50-50 mix of their genes in Molly, effectively. She hasn't got any siblings, and none of her immediate broader family cousins are donors. So what do? You go to the National Register of Donors, look for tissue type. None available for Molly. And so this is what they did. They also elected for IVF and pre-implantation genetic diagnosis. This is in the year 2002 we come to now. And Adam was born. They made 14 embryos by IVF, and only one satisfied both conditions, free of the disease and compatible. And Adam was born. He's going to be a donor, and as I say, the stem cells would normally come from the bone marrow, but we're very fortunate in developmental biology that the cord blood cells act like bone marrow stem cells. It's something to do with the fetal state of them. They're more versatile. And so if you can get the cord blood and it works, then the bone marrow transplant would not be necessary. And indeed, Adam was born and his stem cells from the umbilical cord did indeed cure his sister. And so children who are born this way are called saviour siblings, because they saved somebody who's already been born. Saviour siblings is the kind of phrase that has grown up. But I will say that had the cord cells not worked, and remember it's not a very big population in the umbilical cord, had they not worked, then the next step would have been demanding of Adam, who is not yet old enough to give his own consent, a bone marrow transplant to his sister. And some of you will have read Jodie Picoult's book, My Sister's Keeper, I hope. Has anybody here read it? It was made into a film, and it was on general release last year. Did anybody see the film?
Well, some bought it for Christmas for me, but I haven't seen it yet.
No, don't bother. Read the book instead, because the film is a travesty. But it really talks about the downside. We're kind of focusing forward to a saviour sibling who in the book is now 11, and the downside of being a saviour sibling. And again, you know, I'm asking people to sort of, I'm going to stop here for 30 seconds and just ask you to consider the three cases we've had so far and see whether you think that they are different ethically. I'm not going to ask for any opinions at this point. Okay, so let's move on. I've got one more case, and this one's even weirder. This is Tom and Paula, another Molly. Actually, Tom is called in all the publications Tomato, which is the nickname by which he's normally known. He's a theatre director. But as you can see there, they're both deaf, and the form of deafness is heritable. And their first child was born deaf, which you might expect. The genetics of deafness is extremely complicated. You can be genetically deaf for a large number of reasons. But anyway, it was quite likely that a child was born deaf. But they wanted to make sure that the second one was deaf. And they applied to the HFEA. And the HFEA refused. The HFEA refused because in the guidelines for genetic selection of embryos, it is stated that selection for a condition that is regarded more widely as a disability or a handicap, and I know they're not the same things, but they're there, those words. Regarded as a disability or a handicap by the wider society, you shall not select for. Now, Tom and Paula appealed against that, but the appeal was not successful, This is a story still in progress because she became pregnant without IVF last year, and the baby is soon to be born. So we don't know whether natural childbirth would have brought them another deaf child. So their argument is that in the deaf community, that being deaf is not a handicap, because British Sign Language is now a recognised language, and the community communicate adequately, and in a deaf family, there's no handicap at all in the deaf community. So that's their argument. And again, so we're beginning to move into quite broad patterns or quite broad issues for use of genetic selection. Now, I'm going to put the numbers in context, and obviously Tom and Paula don't come into these numbers because it's refused. That, as I say, about 12,000 IVF babies born in Britain every year. About 200 of those have been born after genetic selection in the way we've been talking about. And of those 200, maybe 2 would be saviour siblings. So we're talking about very small numbers, but nevertheless, they happen. So I'd just like us to just throw this question out. Now, what are the key ethical issues that come in the genetic testing of embryos? And what are the key issues for you come in genetic testing? Something to ponder, something we can discuss in the question and answer session, or maybe after lubrication later. Now, as I said, we know about 4,500 single gene disorders. Some of them are rather vague, but we still see this linkage. And this was the situation at the beginning of the week, where 1,672 tests were being used in some sort of clinical setting somewhere in the world. This comes from the GeneTest.org website, which is run by the National Institutes of Health in the States. It doesn't mean to say they're all used in embryos, and when I say clinical setting, I'm using that extremely loosely, because in many countries, people are allowed to set up genetic tests which actually lie outside the health service provision. And that's true of Britain as well. That for tests that are not to be used on embryos, I can set up a genetic testing lab in my garage. And many of them are to do with testing for paternity, which seems to be the commonest use. But in theory, I could offer you a test for cystic fibrosis and so on. So of these tests, they're not all being used on embryos by any means. And in Britain, where the situation is very highly regulated, anything to do with embryos has to be licensed by the Human Fertilisation and Embryology Authority, current chair of which is Professor Lisa Jardine, and they have the absolute say, yes or no, whether you can do this. And they've added about 30 from the 70 that we had last year, recently, as they've come on stream. And some of them, I think the worry about the commercial use is that in many cases, the people aren't able to interpret correctly the meaning of the test, an increased chance of, but what is that increased chance of what, and what baseline, and so on. So there's worries about the commercialization of these tests by organisations that aren't directly linked with clinics or hospitals, for example. So there's a concern there. Okay, now I'm going to take a step into eugenics at this point, which has been mentioned already in connection with its the misapplication of evolution, and I'm going to pick up that theme again. Of course, eugenics means good breeding, in the same way as euthanasia means good death, thanatos, Greek for death. Genics comes from the word for origins, Genesis is the same root. And Herbert Spencer wondered whether we could apply evolutionary theory to human society, the survival of the fittest for human society. Francis Galton, who's Darwin's cousin and the first professor of genetics, then formalised the theory. But actually, if we look back at history, it wasn't a new idea. The Greeks thought that this was a good idea. Plato and Aristotle were both, in a sense, eugenicists. They believed that society could be improved if the best people had more children. And indeed, I can't remember which way around it was, I think it was Plato, that said that the best men should have intercourse, sexual intercourse with the best women as frequently as possible in order to generate human society. And of course, it was a misunderstanding, not that they knew anything about genes, but if we put it in a genetic context, it's a misunderstanding of what genes do. It's an assumption that personality and behaviour patterns are somehow written into our genetic code, and we now know that that is not so. But it was also the excitement of discovering evolution and then genes was just too much. So let's see what we can do. And Galton was genuinely worried that people he regarded as being the underclass were having like 6 kids, and all the white middle class liberals were only having about 2. So he thought society was going to go down the drain. And so they encouraged the fitter, those deemed by the white liberal middle class to be the best, encouraged them to breed more, and were trying to discourage the so-called underclass from breeding. In Britain, it was never ever enshrined in law, except for a small number of cases, as we would now say, people with severe learning difficulties have been and continue to be sterilised, but a very small number of people. In other countries, it became enshrined in law. And so although we highlight Nazi Germany as being the extreme of the eugenic movement, these other countries all had eugenic laws. They could demand sterilisation of somebody who was deemed unfit to participate in society. And so tragic cases from the USA that were revealed in a BBC Horizon programme some years ago showed that sterilisations in the southern states went on until the 1950s. And a typical situation might be that a black servant girl working on a big cotton estate somewhere down in Carolina might have been raped by the young man of the house. She became pregnant. She was regarded as morally feeble, and after the birth of the child was sterilised. And if the child was a little girl, later on she was sterilised as well. And the BBC tracked down such a woman who had been the daughter of somebody who'd had a child after rape, and that woman had been sterilised. There was classist and racist elements, and also related to things like learning difficulties. And in Sweden, the last eugenic sterilisation was carried out in 1974. So we're talking about people trying to improve human society by preventing some people breeding. That's the kind of nub of eugenics. And the question to ponder, it's been raised by bioethicists since we started to, if you like, select and deselect embryos on genetic grounds. Is there a possibility that we're starting to kind of move into eugenics as we select on genetic grounds? Is it possible that I've decided that this genotype, actually I don't want my baby born with this? And the question will become more poignant as we think about further possible applications. And the further possible applications are these, and what else should we be allowed to select? Remember, it's not just human diseases we know a lot about. We're beginning to, as we start to unpack the human genome and relate the particular variants to particular individuals, we begin to see other features. Where are the boundaries? If I do the bottom line first, what about genes that affect our appearance? Currently, our knowledge of eye colour genetics is it's complicated. But we've got, more or less it's right to say, that the dominant side is the brown range and the recessive side is the blue range. But there are so many modified genes, and it's not that straightforward. And yet, we currently could get your eye colour right with a 70% chance of success. And that's pushing up and pushing up all the time. I reckon within 5 years we'll be up at about 80% chance of being able to select for eye colour. And I was looking at a documentary on the CNN website recently where a San Francisco doctor, genetic doctor, said that once that became that higher frequency of success, he would offer that to prospective parents to undergo IVF and PGD for eye colour. And we can think about similar selections for hair colour and skin tone and so on. And we're also beginning to unpack the genes that contribute to stature. Intelligence is much more complicated because all the social factors are involved. But we can also start to unpack some elements of sporting ability. For example, we know a little bit about the genetics of muscle fibre composition and the fact that Paula Radcliffe has got a very, very different muscle fibre competition from Christina Harugu, a sprinter versus a long distance runner. And we begin to understand the genetics of that. So we could start maybe designing athletes for the 2024 Olympic Games by genetic selection. He's a sprinter, he's a long distance runner. Of course, you've got to make them a runner as well. These are the sorts of things we're talking about. And I wonder where you think the line should be drawn. How do we decide where the line should be drawn? Between selection against severe genetic diseases, selection against rather less severe genetic diseases, selection against things which have got nothing to do with health and disease at all. Where are we going to draw the line? Now, in those of you who are philosophers and ethicists will know about the slippery slope argument, and I'm not a great advocate of the slippery slope argument. I think in most cases it doesn't work. This is the one case in the whole of bioethics where I think we are actually on a slippery slope. And it really is a question of where do I hang my ice axe? Where do I decide to dig in? Okay, very briefly then, for the last few minutes, I'm going to look a little bit at the stem cell thing. Now, I wish this would work, it doesn't. Okay, so I've talked about how these early embryo cells are non-determined. But once we get to this stage, this is in about 7 days, and in a normal kind of migration down the fallopian tube, this little structure, it's called the blastocyst, will be ready to start a pregnancy. Only about 30% of them will succeed in starting a pregnancy. The wastage rate is extraordinarily high in humans, so we have a species that is fertile the year round, and yet after fertilization, only about 30% of the fertilized eggs actually establish a pregnancy. Strange situation. But what we have now, we've got some division of labour. We've got an outer layer of cells which will form the supporting tissues of the embryo, the placenta and so on. And then we've got the inner little group of cells, the inner cell mass, which will form the embryo proper. That will cling onto the surface of the womb and then develop the kind of supporting tube that enables it to feed the placenta. So this is going to be the embryo and then the fetus. Fetus is a medical term. Biologists tend not to use it. You very rarely hear us talking about mouse fetuses. We talk about embryos to the moment they're born, but for human development, doctors have invented the term fetus to describe the growing organism once limbs have started to form, about 8 weeks. So this will form the baby. And what that means is that inevitably it must be true to say that in those cells which still don't have a specific function, except they're going to form the baby, those cells must contain the potential to make every single type of cell in the human body. Every single one. That's the key thing about them. They're going to do that. They're going to make 220 cell types and all the different tissues and organs that make us up. That's what they're going to do. And so we jump to stem cells. Stem cells are cells which can be maintained in a dividing state and then can be persuaded to grow into many types of cell. Well, I've just described it, haven't I? It's the blastocyst. That's what happens in nature. It starts in a state of division and then divides up into many cell types. And the idea is that we might be able to use such stem cells for repairing ourselves when our tissue degenerates through illness or through accident or old age or something. So that, if you develop severe heart disease because of degeneration of the heart muscle, you might be able to repair that. If, like Christopher Reeve, you have a high break to the spinal cord, you might be able to repair that, and so on. And so the idea comes then that human stem cells could be used in human tissue repair. And of course the blastocyst is the obvious place, because those cells in that little mass that hangs inside the skin, that blastocyst, incidentally 0.15 of a millimetre across, and it can start a pregnancy. Isn't that incredible? Absolutely amazing. That's the start of pregnancy. But anyway, that little mass of cells. So it's got everything you need. And so the technology then has been developed that embryos that have been left over from IVF, and in the average IVF cycle, 80% of the embryos are not used. So embryos left over from IVF have been burst open, and those cells have been cultured in the dish and then the way they might develop into different types of cell has been investigated. And so that's a bit like that. It's interesting. I've lost all my colours here on this stage projector. There's supposed to be a blue line at the top and a red line at the bottom, but never mind. But I mean, that's what happens in nature in a sense, very simplified, but this is what we hope will happen, or what it has been suggested will happen in the lab and in the clinic. You culture the cells from blastocysts, you don't allow them to go on to make an embryo, but then you induce them to go down particular lines that are required to mend a particular situation in a patient. And as I said, they've been produced by IVF, and once again, the Human Fertilisation and Embryology Authority are the people that regulate that, and they've given licenses to a limited number of clinics and labs to work on this. The nearest one to here that works on embryonic stem cells is in King's London. Here in Cambridge, they've got an amazing work on adult stem cells. Totally astonishing, but that's a different story. Another embryonic stem cell lab is in Sheffield. But let's go back to the embryonic stem cells. I talked about, in relation to Molly and to Adam, about tissue rejection, and the same would be true of stem cells, that there'd be no reason why, just given stem cells from any old embryo, why I wouldn't reject them. The same thing applies. And so, why don't we try to make clones of a patient? If I make a clone of a patient, and you've all heard of Dolly, you know how that works. If I don't make a clone of a patient, and you do get it therefore to the embryo stage, but you don't put the clone into the womb, you burst it open and you use the stem cells. And what you'd have there, if you cloned a person and took them as far as that 7-day-old embryo, you would have in theory stem cells which were compatible with the patient, and therefore could be used for mending the heart muscle or the break in the spinal cord. And a small number of cloned human embryos have been produced in Britain, one in Newcastle, for example. In Korea, more have been produced, but unfortunately that finding was completely soured by spurious claims made of further work on those embryos, further work that turned out to be completely made up. They did produce the embryos. But quite a large number of human embryos have been cloned in the United States, mainly by biotechnology companies operating outside the rules that George Bush laid down, which Barack Obama has now lifted. So, and we don't know how many. Some companies have been quite secretive about it. But human embryos, cloned human embryos as far as the blastocyst stage, are being produced in different places in the world, with this idea of so-called therapeutic cloning, turning not to reproduce a baby, but to have therapy. And you know, this shows that it does work, that you can clone mammals. Humans turn out to be extraordinarily difficult, as indeed all primates turn out to be extraordinarily difficult to clone. Farm animals, particularly sheep and cows, turn out to be easier. But that's Dolly soon after birth, a couple of weeks after birth, when she's a steady lamb, and with her surrogate mother. And here she is, that's in July 1996. Here in February '97, and that also shocks me, it was so long ago, giving her press conference. Apparently she was a lady of few words. But I've talked with Harry Griffin quite a lot, the Deputy Director of the Ross Institute. He said he'd never, ever seen so many non-scientists on his site. And I was talking last week with somebody who worked in the next door institute, the Forestry Institute, and they were just astonished at these droves of coaches and cars and motorbikes and cameramen arriving. What on earth's going on in there? So this raises more questions. You know, does our motivation, whether we're humanist, whether we're Muslim, whether we're Christian, whether we're Jewish, does our motivation to heal allow us to create embryos specifically as sources of stem cells? And what ethical tools or principles can be used to help us here? And at this point, I think it's nice to have a little bit of a pause to think about ethical frameworks. Many ethical frameworks have evolved or been developed or been revealed, depending on your viewpoint, to enable human societies to work and flourish, to enable human beings to get on with each other. And we all have our own particular framework, and my own claim is that I participate in the virtue ethics framework, But how do we apply these frameworks to these situations? Is it a question of, we suddenly have to think that the human embryo is part of our moral community, or isn't it? And that's a key question. Is the human embryo part of our moral community, and therefore worthy of moral considerability, whether we're working in a consequentialist or a deontological, a utilitarian or a virtue framework? What sort of ethical framework can we bring to bear to make decisions about embryos, whether we're genetically selecting them or whether we're using them to make stem cells? And one of the things that it hinges on in both areas is, as I've hinted, the ethical stroke moral status of the early embryo. And I'm just going to give you the two views at the opposite end of the spectrum. Completely opposite ends of the spectrum. There's everything in between. The first is the early embryo is a human person. I was giving a talk at a Methodist church in their cafe-style church recently, and I was just talking about genetic selection, and somebody stood up and said, the Bible tells us that life is sacred from the moment of fertilization. And I asked him to point out where in his Bible it said that, because it doesn't. Fertilization was unknown in biblical times. People didn't know that women produced eggs until the 18th century. A woman became pregnant when she missed her period. That's when it all started. And it was the man that provided the seed. So we cannot say the Bible teaches us that life is sacred from the moment of fertilization. Anyway, fertilization is not a minute, not a moment, it's 10 hours. So if we believe there's a biblical guidance here, it can't be based on specifics. It needs to be based on more general application. And of course, at the other end of the spectrum, you have the early embryo is not a human person. But we can't ascribe human personhood to the 1 cell, the 8 cell, the blastocyst stage. But we don't ascribe human personhood until rather later in development. But of course, everybody who holds that position will equally acknowledge that at least that embryo has the potential, in appropriate circumstances, attached to the lining of the womb, developing into a fetus and coming to term, to become a human person. And I find it very interesting that Christians find themselves in different camps. And I've heard the love your neighbour argument applied by both groups. And so my friend Philippa Taylor, who works for CARE, has written a little book about protecting the weakest of our neighbours. And the weakest of our neighbour, in her view, is the 1-cell embryo. And I've also talked with and met Ted Peters, the American theologian, who believes that loving our neighbour imposes on us a mandate to heal those. Adults are already here, children are already here, babies are already here. It imposes on us a mandate to use whatever means we can to heal them. And in his view, stem cell technology is completely acceptable. And in fact, his latest book is called Sacred Cells, why Christians should support stem cell research. And he also uses the neighbour argument, which is very interesting, isn't it? So they both use the same argument. And of course, the particular way that we work out our worldview, our philosophy, will affect what we think about this. The particular way we work it out. And I'm just going to very quickly talk about, if you like, what my friend Philippa Taylor would call ethical stem cells, which is a value judgment on the grounds that all others are unethical. Adult or non-embryonic stem cells are located at specific sites in the body. Their function is to replace our cells as we continue to turn them over and to help us to repair ourselves when we're injured. And so skin stem cells, the fibroblasts at the base of the skin, are working all the time as our skin turns over. Some stem cells are less easy to activate than others. The stem cells that are to do with repairing the central nervous system seem to be very hard to activate, even when you've had a severe brain injury. And the group here in Cambridge, the Brain Repair Centre, are doing incredible work on that. But of course, in nature, these are limited to a small number of cell types. The bone marrow stem cells are constantly replenishing our blood cells, but they don't replenish our muscle cells, as an example. But in fact, the history of adult stem cells turns out to be very interesting. We would think that in vivo, in life, because they've got a limited potential, they won't be very useful. If you can culture bone marrow stem cells, but what are you going to do? Are you going to try to use those to repair heart? But after a lot of false starts and a lot of data which were open to other interpretations, We now know that under some circumstances, adult stem cells can be persuaded to change their career pathway and go down a different route. They transdifferentiate. And some clinical trials are now underway. And again, exactly why some patients are responding to injections into their heart of bone marrow stem cells is not at all clear. Biologically, it's not at all clear. But it is clear that some patients are responding to this and others are not. And so it may be that some adult stem cells do have the potential to repair damaged tissues. And then we've had all sorts of other sources, stem cells from testes, and men cross your legs at this point. You can make stem cells from cells that are on their way to making sperm, but before they've done that cell division that reduces the chromosome. And then I've talked about cord cells, and the group in Newcastle succeeded in persuading cord blood stem cells to develop into cells we call hepatocytes. They're going to make liver, and they made a tiny liver-like structure the size of a penny out of cord blood. But the most interesting, and I think has probably changed the ethical climate here, was that two groups, one in Japan and one in California, used those skin stem cells I talked about and persuaded them by a little bit of genetic modification to behave like embryonic stem cells. And it was very exciting. Two different groups, and of course the papers were published at the same time, back to back. So these are called induced pluripotent. Pluripotent means they can do lots of things. Induced pluripotent stem cells. And I think those of us who've been talking about the ethics of stem cell technology at this point think the ethical climate has changed, because if you can do this with skin fibroblasts, turn them into embryo-like stem cells, then clearly that obviates the need to use embryos. It has a further advantage, is that you can make them patient-specific. So my stem cells from my skin could be used for repairing me. And then, just before Christmas, an American group managed to persuade these induced pluripotent stem cells to actually change into nerve cells in the lab. So we're beginning to see everything that's hoped for in embryonic stem cells, but the research has been going on now for 9 years, and still no therapies have been developed. We're beginning to see the situation taking over. Now, I will say that there has been a little bit of cynicism expressed about this. The chairman of one of the biotech companies working on embryonic stem cells says, you'll never get a license to do this, because each cell line will need its own license. But my own feeling is that where there's a will, there's a way. That surely one can have a blanket license for this type of treatment. So, will then these technologies eliminate the need for embryonic stem cells? And I think, obviously, if you're opposed to the use of embryonic stem cells, and I'm not making any assumptions about what you think, and please don't make any assumptions about what I think, then obviously you would hope so. So it is a question of watching this space. And so let's go back to a question I posed earlier. Does our religious faith help us to make ethical decisions in these areas? What form does that help take? And in both of the major faith groups we have in Britain, there is disagreement within those faith groups. I would say amongst the Jewish community are mostly okay with all these technologies, mostly, except for the really orthodox Jews. So that's a big question. What form does our faith, help from our faith, take in helping us to decide? But here's a few resources that you might want to. I think my paper is Faraday paper number 7, it might be 8. And then Ethics in Brief is published by the Kirby Lang Institute of Christian Ethics, based at Tyndall House on the other side of Cambridge. Ethics in Brief is all about, my commentary, ethical commentary on the new embryo bill. And there's this book, which is, and this is my very ancient book, going back to 2004, is on the bookstall. Some of the science is out of date, but the principles that we kind of try to discuss, I guess, are still up to date. And I hope it's there at a very cheap price. So I'm going to stop there, Mr. Chairman. I think it's time to stop.
Thank you, John, very much. That great overview. And I do recommend Life on Our Hands. And of course, like all our other books, it's heavily discounted. I hope it's been pointed out, by the way, thinking about books, that we do try and we don't, we just buy books and sell them on pretty much cost price, just enough to cover postage or whatever. So I think you'll find all the books there are quite a bit cheaper than you would find them out in the other places. Even Amazon, we try and undercut Amazon whenever possible. So. Okay, so over to you for any questions, and I'm sure that stimulated plenty of questions. So.
Stunned silence. Yes.
Please.
Yes. Hybrids was mentioned. I was wondering what that is. I haven't seen it before. Sorry.
Okay. Yes. I decided that although hybrids was on the slide, I took a quick look at my watch and thought, no, we can't do that. Hybrids. If you think about what you need to establish a clone, you need a donated egg. Had I been giving a different lecture, I'd have talked to you about the shortage of human eggs. Egg donation is not a straightforward procedure. And so persuading people to donate eggs for research, let alone for fertility treatment, is obviously quite difficult. So there's a genuine shortage of human eggs for fertility treatment, and there's an even stronger shortage of human eggs for making clones, because you have to take a human egg and you have to empty it of its genetic material, and then put in the genetic material from a pre-existing person. That's how you make a clone. And so an egg is to be donated that's going to be effectively just a vehicle for DNA. And so it was proposed that instead of using human eggs, we might use eggs from other mammals, and rabbits and cows were considered to be appropriate. And so licenses were granted to empty cows' and rabbits' eggs of their main genetic material and to replace that with human genetic material to start the cloning process off, just to make stem cells. Now these are cell hybrids, and cytos, the word for cell, and hybrid, and so they're called hybrids. And as I say, there are 3 labs in Britain that have got licenses to do that. We hear very little about it after the new developments.
Other questions? Please, yes.
Mentioned these differentiated stem cells, and then basically encourage them to turn into what. How do you determine what they turn into? How do you make them go a certain route?
Yeah, well, this I think is if you like the sticking point, because if you leave embryonic stem cells and don't take any steps to keep them youthful, and you've got to keep the youth genes switched on, they will start to differentiate. But because they're not in their normal bodily niche, they don't differentiate into a kind of balanced package of the different cell types. They tend to go in one of a few directions. They do tend to go towards fibroblasts, for example, the stem cells that we find, for example, at the base of the skin. And so diverting them from things that they want to do to things you want to do turns out to be difficult. I had a student doing a project on stem cell technology, and I sent her down to Stephen Minger's lab to interview him. And she asked him, this was not long ago, is it possible to get a range of cell types on demand? And he said no. But what I found interesting was that group who've induced differentiation in this new stem cell lines had actually got them to make nerve cell precursors, which has not been achieved with embryonic stem cells. That's quite interesting. So getting the cells you want is the tricky bit. And there's 6 cell types that you will find in the average culture if you just let them go. And of course, one of the things that you're worried about is one of those cell types might be a carcinoma type cell.
Yes, please.
Part of our human makeup is how we respond to disability, disease and suffering. With this sort of experimentation that you're doing, aren't you in danger of actually changing the human person forever?
I think there are two different elements to your question. The first is actually to human suffering, and I entirely agree with you that it is a mark of a humane society that we visit the sick, we clothe the naked, we visit the prisoner, we care for those who are less able. That is a mark of humane society, and in fact Jesus said it's a mark of the kingdom of heaven. And I think anything that would diminish our regard for people with disabilities would be a bad thing. And it is true to say that if genetic technology of the genetic selection sort that I've talked about was applied right across the spectrum of conditions that we know are regulated by genes, we would, in a small proportion of society, be reducing the incidence of particular disabilities. And Tom Shakespeare asks us to think about, how does that affect our attitude to people who are here already with disabilities? And so, for example, a family who already have a child with cystic fibrosis, they don't really want another one, but they take steps to avoid it. But how does the child who's here already feel about that? So there's that element as well. But the second element to your question is, are we changing the human species forever? And I think we need to realise two things. Firstly, this is not mass technology. The genetic selection procedures I've been talking about do require IVF. And anybody who's undergone IVF, or anybody who knows friends or relatives who have undergone IVF, know that for the woman, and to some extent for the man, for the woman in particular, it's a physiologically and emotionally demanding procedure. And so therefore you need to have a very good reason for doing it. And this comes down to thinking about will we apply it to non-medical situations. So you need to have a very good reason for wanting to do it, not a mass procedure. And secondly, it's a procedure. The genetic testing procedure is very much a procedure of developed societies. You know, we forget that there are 6.7 billion people on this earth, and only a fraction of them have got access to these technologies. When the Human Genome Project first draft was announced, we had Tony Blair on one side of the Atlantic and Bill Clinton on the other side giving their press conference. Bill was talking to students at one of the universities in Washington and saying, you know, this means that you guys are going to live to be 100. But he forgot that actually, you know, the age expectancy in parts of East Africa is 40. So it's not mass technology, even in the West, and for a large proportion of the world's population, it's not available. And that might give us pause for thought as well. I'm going to throw a question back at you, that whereabouts down my kind of ladder or slide of genetic testing did you want to stop? Did anybody want to stop before Matthew, so we didn't do it at all? Okay, that's fine. Did anybody want to stop before Chloe? So the late onset, you know, I'm going to put my hand up here. I'm going to declare a hand here. Did anybody want to stop Adam being born to save his sister? That's interesting. Did you want to stop?
Oh yes,
yeah.
yes.
So we wanted to stop Adam being born?
The thing with the Adam thing was what happened if he wasn't compatible? It was there that...
No, he was bound to be compatible because he'd been tested for his tissue type.
Yeah. It was whether you then had later to take bone marrow from him without his permission.
Yeah, well, without his permission.
Where I got stuck.
Indeed, and how much more would be required of him? And what happens if his sister died and he knew he'd been born in order to save her?
Yeah. Yeah. I mean, is he an instrument or is he using?
That's a good question. So, with regard to those particular cases where it would not allow those particular situations.
See, I'm okay about Adam, but not about Chloe.
You're okay about Adam,
Yeah.
all right.
Even though there's a danger of such children being commodities, I'm okay about it.
Yeah.
I think we are trying to care.
Yeah.
I was going to ask a question, which I'm not sure what to ask, but there may be, you may know of individuals who can have the right to have a child, but we may know that they may not want that child, may not love that child, yet we wouldn't then stop them having a child.
No.
Is that a comparable dilemma?
I was asked to comment on the Saviour Siblings scenario on Premier Radio when the BBC ran that very bad documentary a few weeks ago. And as I pointed out, and you've just hinted at, you know, people have children for different reasons. They have children to save their marriage, they have children to get a council house, they have children to have a fashion accessory on their arm, you know. So having a child to save another seemed to be a rather higher motive than any of those. But I do acknowledge that if we're trying to work it out in ethics of blackness and whiteness, then there's an awful lot of black in that. And this is why I try to say I'm trying to apply virtue here, what is the most virtuous routes, but you may disagree with me on what the most virtuous route is. What about Tomato and Paula with baby Molly? Well, Molly was already born. And he really is called Tomato, you know. He's
Their decision was to have a second child, regardless of whether it was to be deaf or not.
Yeah, they had one. I mean, the genetics is complicated, but they had a very high chance of having a deaf child.
Yes.
It was probably greater than 50-50 because of the genetic complexity of their cases. But
They wanted to ensure it.
no, they wanted to ensure it was, but they then said that they were also concerned that because Paula was 44, she wouldn't conceive very readily naturally. But if that is true, then IVF was less likely to work as well, because IVF is partly related to the woman's age. So all in all, it was a difficult situation for them. The HFEA was absolutely clear-cut about it.
You didn't go to eye colour yet, John?
I haven't got to eye colour. Well, you know, the HFEA, in its general discussion of these issues, says that they will only consider genetic selection for therapeutic purposes, for purposes which relate to avoiding having a child with a serious genetic condition. And they will not tolerate applications for non-therapeutic conditions. In other words, what they call enhancement. So they won't allow enhancement. But it is actually quite difficult, even in medical practice, to distinguish at some points between enhancement and therapy. So, and the cases that my friend Peter Turnpenny, who is the professor of clinical genetics at Exeter, and I talk about when we're talking about this different groups, is we can think of two types of case where the boundary has been fluffed. So Peter knows of a young woman who has had leg lengthening surgery so that she can be tall enough to be considered to be a flight attendant. And that was done on the NHS as a therapeutic procedure. It's a very unpleasant procedure. And then I think, you know, even more so, you have these young women who misguidedly think that they're going to be a better, brighter, happier person if their bra cup size goes up from A to C. Totally mistaken, but they claim that their psychological health will be improved if they undergo this surgery, claiming it's therefore therapeutic and not, as people think, enhancement. So the line is difficult to draw. But I think we should try to draw the line, and that would be a good line to try to draw between something that is genuinely therapeutic, and then there are shades of line drawing, and something that is not therapeutic at all, like appearance. Now, if we were in the United States, we wouldn't be having this discussion except in very general terms, because if you can pay for it in the States, you will be allowed to do it. That's been very, very clear, for example, in the state of California. The people who want enhancing gene selection and are desperate enough to undergo the IVF in order to do that will be allowed to do it.
Well, on that sobering note, I think what we'll do is now we'll break because we want to have 10 minutes before dinner. First of all, let's thank John very much for a very thoughtful and interesting talk.